is Carla Petrie on the presentation of co-immunoprecipitation and immunoblotting. Background and principle of techniques. Co-IP is used to analyze the interaction of multiple proteins and their functions. A protein complex is isolated by co-IP using an antibody for one of the components in the complex. There are certain criteria for the antibodies. They must bind to the surface of the complex, and they use an anti-tag antibodies. Here is a diagram of how the co-IP works on the left-hand side, and the steps for the co-IP. The protein complex mixture to be analyzed. The antibody is added, and the second antibody, or the protein AGB, is used to bind to a specific antibody. There's an incubation phase. It can last from an hour to overnight, depending on the type of protein. The next phase is to collect by magnet or centrifuge the protein. And after you collected the stuff, you need to wash to remove the cellular matter not bound to the antibody. And then the next phase would be to elute the protein on an SDS page to identify the protein that you're looking for, or to do the blotting, the immunoblotting on there. The research article that I found would combine the co-IP with mass spectrometry. On here, identified protein-protein interaction was once limited to yeast 2 hybrid analysis. Now, protein-protein interaction can be examined in both cells and native tissue by precipitation of the protein complex. Neuronal proteins do not exist as sole independent units, but as a large macromolecule complexes interacting proteins. And the figure to the left shows the steps for the CRP with the mass spectrometry. So it's the same thing as just the plain CRP, but they add the mass spectrometry to get a better idea of what's in the protein. And this process could take up to 2 days, depending on full steps, because each step needs to be very efficient steps. So it depends on the proteins that you're looking for. So on the results, so after you complete each all the steps, then so the results depends on the specific bait protein. So here on the left hand side, if the results are visible with the chromatic blue staining, then the same results would be available to see with the mass spectrometry. Say that word. And so you would see the better protein that you're looking for. And the other interaction proteins can be verified by other means, like immunoblotting, like the image here on the right. So some of the strengths of using this experiment is that the adding the mass spectrometric with the co-IP has allowed for the study of protein-protein interactions within the cells. And a few drawbacks with this experiment is that it's very specific for the type of detergent used for the proteins, and the antibody is very specific on which protein you're looking for, and the washing time and condition. Everything is really specific, and and the time it takes for the whole process um, is really specific on the co-IP itself. So my question for this, the co-IP is, do they know if there's any proteins that co-IP can't isolate for, even if there's an antibody for it? Thank you.